Welcome everybody to our webinar. My name is Michael Hannus. I'm the founder and the managing director of Cytus Biotech, and it will be a pleasure to talk to you about RNAI interference and transient gene silencing. The talk will take between like 30 and 40 minutes, and this is what I'll be talking about. So I will start with one single company slide to tell you who we are and what we're living off. And then there will be 4 sections that will also give you an opportunity to join again if you lost me on the way. The first part will be on why RNAI is such a useful technology. And the second part, I will introduce the problems of RNAI that have to do with off-target effects and a mysterious sequence called SEED. In the third part, I will talk about how complex pools of siRNAs can overcome this problem. And in the fourth part, I will show you how, in fact, these SEED effects can be useful. So here's the company slide. Cytus Biotech is a relatively young company. We are based in Munich, Bavaria, Germany, and we are mostly an RNAI company. So our first and main product are the siPOOLs, and this is what I'll be mostly talking about. These are complex mixtures of siRNAs. In addition, we have RNAPools, these mixtures of probes used for RNA pull down, for RNA biochemistry, and then a more recent product of RiboPools. Those are tools to deplete ribosomal RNA for RNA-seq, so you don't end up only sequencing ribosomal RNA. And lastly, we have services, some of which I'll be talking about, something called PhenoVolt. And then in addition, we offer RNAI experimental services, knockdown validations, transfection optimizations. And in fact, we'll even run an RNAI screen for you if you need our help. Let's jump right into the science. And the first part, where I would like to convince you why RNAI is such a cool method. So if you compare RNAI and CRISPR, it's actually quite interesting. So you may remember that both of those were revolutions in the last 20 years of functional genomics. RNAI about 20 years old and CRISPR about 10 years. And they're similar and dissimilar. Both of them require short RNA molecules to give a nuclease specificity. In the case of RNAI, this small RNA is called siRNA, binds to a protein called argonaute, and this then functions exclusively on an RNA level, binds RNAs that have the same sequence, that are complementary, cleaves that RNA, and then the RNA gets degraded. And if it's a coding RNA, there's no protein. So for CRISPR-Cas9, the small RNA, as you know, is the sgRNA or crRNA. It again has about the same length, 20 bases, and it drives the specificity of a nuclease called Cas9. Now here comes the first fundamental difference in that argonaute, nuclease in RNAI, is in every higher eukaryotic cell, whereas Cas9 is a bacterial protein, so you have to bring it into the system. That acts on a DNA level, on a genomic level, as you know, causes double-strand breaks, which then causes gene repair and in some cases introduction of mutations, frame shifts, and for instance, truncated proteins or other effects that you will probably be aware of. So again, they're similar in that they both use small RNAs to drive specificities of a nuclease, but they're different in that for RNAI, all you have to bring in is the siRNA, and for CRISPR, it's both the nuclease and an sgRNA. One is on the RNA level, the other is on DNA level. Actually, we had, so now in this, what I would like to show you are 3 use cases, or 3 case studies of RNAI. And the first was actually funded by the Bavarian government, and in a grant where we were comparing RNAI and CRISPR reagents and technologies. And in the slide that you'll see here, we're actually focusing on 2 genes, lamin A and EMD, and we picked those because they're very good antibodies for those and very clear staining patterns. So you can actually see the expression of those 2 genes on each individual cell. So on the left side, you see in an RNAI experiment And hopefully you can see the mouse. So basically, on the top level, you see it. So basically, we stained cells for two different things. For three, we stained the nuclei with DAPI. We stained for lamin A and for EMD. And we're knocking out, knocking down with an siRNA, lamin A and EMD. And what you can see in the central panel of the left side, where lamin A was silenced by a lamin A siRNA, you see basically that uniformly all the cells lose the lamin A staining. This is here. And in the lower panels here, where EMD was silenced, you see equally that basically all the cells lost the EMD staining. That would be the screen staining here, and there's none left. Now, what that means is that basically RNAi affects all the cells more or less homogeneously. On the right side, you see a corresponding experiment, a CRISPR experiment, where we actually use two sgRNAs targeting lamin A here in the center and the bottom row. And here the situation looks very different. So actually at a first view, you basically don't see any cell that's affected by the CRISPR knockout. And you have to look very closely to, in fact, see two cells. That's where the arrows are pointing in the central row, and one cell in the bottom row, where indeed lamin A was edited, was knocked out in a cell. So we did this experiment also for other genes, for MD, for actually other genes with multiple reagents, multiple also transfection reagents, approaches, and the result was always the same. And the efficiency, at least in the cell lines that we tried, was a low single-digit percentage. So what that means is that if you run a transient CRISPR assay, you need a very sensitive assay to actually pick up 1 to 2% edited cells, or actually you have to do cloning. Now, the second case study comes from a collaborator of ours, Michaela Bauer, who is a predoc working at Gunther Meister's lab at the University of Regensburg. And Gunther and his lab, they're interested in T-cell infiltration in cancer. So you may know that cancer prevents the infiltration of T-cells to not have the immune system attack the tumor. And Gunther's lab is studying the genetic background of that, and they would like to run an RNAi screen, to which end Michaela has been bravely trying to establish or working on an assay since 2 years, which is very complicated because in fact she has two cell lines. She has to work with T cells and she has to work with cancer cells. This assay requires automation, transfection, live cell washing, and for a decent screening window, requires search for an optimal cancer cell line and for positive control genes to set up the assay window. Now, the problem is that cancer cells have developed redundant escape mechanisms to avoid T-cell infiltration. So knocking down just a single gene is not sufficient. In fact, from the literature, Michaela found out that she has to silence 3 genes at the same time. Beta-catenin, STAT3, and ATF3, where at least beta-catenin is an essential gene. So what you see here on the left side, and upper left, is an experiment where Michaela knocked down these 3 genes individually, one after the other, with one siPOOL each. And actually, these siPOOLs, or these RNAi reagents, were provided by siTools. And what you can see is that you get pretty decent knockdown, best for beta-catenin, and also good for the others after 48 and 72 hours. So that's, again, with individual siPOOLs. Now, on the right side, she did the same, but she actually mixed the siPOOLs and silenced the 3 genes in one experiment. And what you can see that at least in the lower half, and after 72 hours, you see that the transfection efficiency, the knockdown efficiency, is actually pretty much the same as you get if you work with individual siPOOLs. So what that means is that you can knock down at least 3 genes at the same time if you just combine the RNAi reagents, which is very convenient. There's a second thing that I would like to point your attention to, and that is the concentration. So she's actually using 1, 3, and 10 nanomolar, and you can see that the maximum knockdown, so here the maximum knockdown is actually reached already with 1 nanomolar total anxiety concentration. So what that means is that for each gene, she's basically using 300 picomolar. So the siRNAs or the siPOOLs that we're providing are extremely efficient. The third case study I had to show you, that is showing how RNAI can be used to silence a virus. In fact, our favorite virus, SARS-CoV-2, in viral cells. So these images come from a collaboration that we have with Stefan Finke at the Friedrich Loeffler Institute of Reims, who are actually allowed to work with SARS-CoV-2 viruses and with whom we have an excellent collaboration since about a year. So what we've been doing here is silencing or transfecting cells with different siPOOLs and then infecting them with the virus and seeing how much that affects viral infection. So on the upper left, you see a cell lawn transfected with a negative control siPOOL. Blue are the cell nuclei, green are the infected cells. And what you can see is that pretty much all cells are infected. KIF11 is just an RNAI control, and then you have ACE2. So ACE2, as you probably know, is the SARS-CoV-2 receptor. And even though we know that the knockdown is actually very good, you see that it doesn't really reduce infection much, which probably means that the virus needs very little of this receptor to still get into the system. Now at the bottom, you see 4 different siPOOLs that are just called SARS-1, 2, 3, 4. And those target different regions of the virus itself. And what you see at your first glance is that here, the efficiency is partly very good. The siPOOL that are called SARS-3 basically almost completely blocks infection in these cells. If you look well, I think like 3 cells left that show some infection. What you also see is that depending on what region you target, the efficiency is different. So I think both of this is interesting. It matters which region you target, and what you can see that RNAI is in fact able to pretty much block a virus that would infect every cell. And obviously this is something we are very interested in and that we're following up on. So what I wanted to try to convince you about in this initial part was that RNAI is cool because A, it's fast. An experiment takes just days, not weeks. All you have to do is you have to transfect in a siRNA in whatever cell line you're working with, as long as it's transfectable. The effect that you get is very penetrant and homogeneous, which is cool because you can expect with a cell population, pretty much every cell will see the siRNA. It's a knockdown, not a knockout, which allows you to work with essential genes. And it's transient, which has several advantages, one of them being that you have no adaptation. It's something I didn't talk now about, but it is a problem knocking out genes that cells will adapt to the absence of a gene. So there's a whole bunch of reasons why transient knockdown with RNAI is actually very useful. And basically what that means is RNAI overall is pretty much a drug-like molecule. So now, after I've been praising RNAI so much and telling you how cool it is, I have to admit that RNAI has a problem, and that is the off-target effects, which is the second part of my talk. So when RNAI was discovered 20 years ago, the excitement was enormous. In about 2001, when Tom Tuschl found the siRNAs, first experiments seemed to indicate that these are extremely specific. So basically, if you took 1 or 2 bases, modified them, you would not silence your target gene efficiently anymore, from which people concluded that the siRNA is extremely specific. I was actually then joined, in 2001, I joined the first RNAI company called Phoenix Bioscience, and we were amongst the first to produce RNAI libraries and actually also to screen them for industry clients. And we noticed relatively early that there were problems with it, but as the excitement for RNAI was about as enormous as it is now for CRISPR, no one really wanted to see this and publish it. It took about 10 years for people to get to publish this paper. And I think this is very interesting, which is why I would like to walk you through this figure. So this is presenting the data from a genome-wide RNAi screen. For the matter of this discussion, it doesn't matter what the assay is about. It's wingless signaling, but it doesn't matter. You see basically a normalized readout on the Y and the X axis. of these scatter plots. So in this first plot, you see each point is one siRNA in this genome-wide screen and tested twice, replicate 1, replicate 2, and you see a scatter plot comparing these two results. If the results are identical, it ends up on the diagonal between the lower left and the upper right. And the R value is 1, the correlation coefficient. So what you see is if you repeat an siRNA in a good assay, you get the same result, which means these siRNAs are reproducible. Now, it's the same screening data, but just plotted differently. Now here, you have each point being two siRNAs that target the same gene, just with different sequences. And what you see is just the opposite. So you see that basically the R value is close to 0, and there's no correlation. You basically see a spherical cloud here. So what that means is that globally, two siRNAs targeting the same gene will normally not give the same phenotype. Not always, but most of the time. Now this plot, the last plot, brings a bit of reason to that. So what is driving these differences in phenotypes? So here the same data is again rearranged in a different way. So here each spot are two siRNAs that have the same seed sequence. So what is seed sequence? The seed sequence is the sequence from base 2 to 7 or 2 to base 8 of the antisense sequence. So it's a 6 to 7 mer sequence. And so what you see here is that if two siRNAs have the same seed sequence, they will have a similar phenotype. The R value is 0.53. So what that means is that globally, the 6-mer sequence is more important than basically the matching to the target sequence. Now that, of course, was a shocking result. However, people kind of should have anticipated it, because this is a paper from 2003 by Amy Jackson, and she transfected in this RNA against MAPK14 in different concentrations, 100 nanomolar, that's on the top, and 160 picomolar at the bottom. I think it was HeLa cells, and then she ran expression arrays, Affymetrix arrays, because there was no RNA-seq then. And what you see here on this panel is basically downregulated genes as green stripes and upregulated as red stripes. So what you see here, the green stripe here, that's the target gene that you should downregulate, and you see that in fact it's a green stripe. But you see that there are many other genes that are deregulated, and some actually upregulated. And you have to go down very far with the concentration to get rid of these off-target effects, but then also your target gene silencing becomes weak. So there is no concentration where you get good target silencing, but no off-target effects. It's important to keep in mind for the rest of the talk. So what's the molecular background behind this? So initially when RNAi was discovered, people only saw this first. So that's what we call the canonical RNAi pathway, where an siRNA binds argonaute, and then argonaute will cleave a matching mRNA at base 10, and it leads to cleavage and then degradation. That's what I had explained initially. However, there's a second pathway, and that only requires, and that's basically the microRNA pathway, or the endogenous RNAi pathway. So there are only 6 spaces in the seed region matching to a target mRNA, or RNA in general, are required to cause dealylation and degradation of the message, or of the RNA. So now, if you do the math, if the siRNA is well designed, it will only hit 1 gene with 19 bases. But with 6 bases, it will theoretically target thousands of messages. Now, is that really the case? And again, that also has been studied by a very interesting paper in 2011 by Garcia et al., also published in Nature, and they had been looking at several siRNAs and microRNAs and quantified the number of deregulated genes, that are the genes that are downregulated, by at least 2-fold using Affymetrix arrays. It was a very expensive experiment. And here you see the numbers, and basically you see a distribution. Some siRNAs will actually deregulate up to 700 genes, whereas others are completely clean, and you have everything in between. If you do the statistics here for microRNAs, and there are actually with some microRNAs in between. So if you use so microRNAs and siRNAs as a ballpark figure will deregulate around 100 other messages in addition to their target gene by a factor of 2. Now, of course, that's a problem. And by the way, it doesn't have so much to do with what reagent you use. So this is data that we derived from several published RNAi screens with all the different libraries that are commercially available. And we basically calculated the correlation coefficient between siRNAs targeting the same gene. And what you can see is that this is in these box plots, that it's basically close to 0 for most of them. And just as an interesting point, the on-target plus from QIAGEN, which have strong chemical modifications and which should have no off-target effects, those actually have a very low correlation coefficient, so they show plenty of off-target effects. So it's basically something that you cannot avoid by design or by chemical modifications. Which brings me to the third part of this talk, that is how we approach this off-target problem and using our complex siRNA mixtures. So the existence of seeds was actually discovered in 2006 by Dharmakon, which was an excellent company then, ran excellent research and published great papers. And they were touring through the world with a slide deck showing the following slide that I'll be presenting you. So they did the following experiment. They generated 64 siRNAs against a target gene. And then they generated pools of this. So they made 16 pools of 4 siRNAs, 8 pools of 8 siRNAs, and so on, and 1 pool of 64 siRNAs. And then they were testing those for specificity and knockdown efficiency. So this first slide shows you the effect of efficiency. So here in this, what you see here is the siRNAs, the single siRNAs. And on the y-axis, you see the number of deregulated genes measured by Affymetrix arrays. Again, very expensive experiment. And so what you see is actually exactly the same that I was showing in the previous slide. You see everything from very clean siRNA, showing just a few off-targets, to those that showing 200 off-targets. And the average is pretty much the same at 80 genes being downregulated on average. So as you now pool these siRNAs more and more, 4, 16, you get less and less off-target effects. And if you pool all 64, you're down to less than 20 deregulated genes. So just by pooling siRNAs, you get rid of these off-target effects. And just for completion, I also want to show you this. This is doing a similar experiment, just here now not measuring the number of off-target genes, but basically measuring the efficiency of the knockdown that's shown here. This is basically knockdown efficiency. Bottom is 100%, and this is no after. And so what you can basically see is you have siRNAs that basically have perfect knockdown up to basically no knockdown, and the average here is about 40. And then again, as you pool them more and more and more, your knockdown gets better. And basically, if you pool all 64, your knockdown is as good as basically your best siRNAs. So that again shows if you want decent knockdown or very good knockdown, all you have to do is just pool many siRNAs, but you really have to use many. And that basically brings us to what we basically call the siPOOL concept, even though I have to give credit to Dharmakon, who came up with this first. And it's depicted in this figure. So on the left, what you see is that a single siRNA here, I hope you can see the mouse, a single siRNA on the left will, if it's well designed, have one target siRNA. It will target basically one target gene. But it will inevitably have many off-target effects, and unfortunately, it's very hard to predict what these are. And I told you it will be around 100 normally. So all you have to do is you have to pool many siRNAs and design them such that each of them has a different seed region, such that the concentration of each siRNA is very low, and such that the off-target effect caused by the seed sequence is basically diluted out. So you ran experiments in the early days of the company to see if that's indeed true. And what we did here was a spiking experiment that we actually also published. So we took one siRNA from a publication by King et al. In 2012, of which we knew that even at low concentration, it gave a very strong off-target effect. And that's here depicted by this red bar. And we then combined this single siRNA and in pools with other siRNAs, targeting having the same on-target gene, so pools of 4, pools of 16, 30, and 60 siRNAs. And then, with actually 4 different assays, reporter assay, RT-PCR West, and the functional assay, we only probed for the off-target effect of this one dirty siRNA that was known, right? So we knew that it had an off-target, and we only looked at the off-target effect. And this is the data that we got, and I'll walk you through that. So we made a luciferase. I just show you for the reporter assay. So basically, we made a luciferase construct. We fused luciferase to the 3' UTR of the gene MAD2 that's causing this strong off-target effect. So the off-target effect in this assay becomes obvious in the downregulation of luciferase. And then we ran this assay here, and where we transfected each single siRNA in pools in concentrations of totally 1, 3, and 10 nanomolar. So relatively low, right? This is the control, the negative control siRNA. And this is here now just our one single dirty siRNA, transfected 1, 3, and 10 nanomolar. And you see a 60% reduction of luciferase activity. And here comes the interesting part. If you now combine this with 3 others, the off-target effect here gets reduced, but only a tiny bit. So maybe like 10%. It's still a strong off-target effect. Now here we have a pool of 15. And you see that most of the off-target effect is gone, and when you go up to 60, it's completely gone. So what that shows, and that's actually a very important message, is because actually that's frequently used. These low complexity pools, just pooling 3 or 4 siRNAs, will not help against strong off-target effects. The single siRNA a single siRNA that has strong off-target effect will still have a high enough concentration that it shows a strong off-target effect. You need complex pools to get this dilution effect. So here we show this with another experiment, so with a classical expression analysis. So we took, again, the same dirty siRNA from this publication and the previous figure, and we transfected it 3 nanomolar, so again, very low, in HeLa cells, and used mock transfection as a control. And here you see a classical MA plot. Each point here is a single gene, and you see the y-axis is basically deregulation as compared to the mock control. And here's just overall expression level. So again, what you see here, the green spot down here, where it says target gene, that's our target gene. And you see there's a more than log 2-fold downregulation. So target gene is downregulated well, but you see there are many, many other genes that are downregulated, and as a secondary response, also then upregulated. So it's basically a transcriptional mess that's caused by this siRNA. And it would be blunt to claim that whatever phenotype you're seeing in this cell is only the effect of silencing your target gene. The effect that you'll be seeing is probably a mixture of all the from all the genes that you're deregulating. Now you take this very siRNA and you combine it, you put it in a complex pool of siRNAs, and your target gene is still downregulated well, but most of the off-target effects are gone. And you see a very nice separation between the genes that you do not want to target and the gene that you want to target. It's, again, we're still doing biology, right? It's not 0. You still have a few genes that are derelated by 50%, but it's far less, and there's a very nice separation. So what this shows is that even a very strong off-target effect from a very dirty gene, from a very dirty siRNA can be removed if you put it in the surrounding of a complex siRNA pool. Now, I think I mentioned initially, and that a very critical point is the concentration of the siRNA. This target dilution, it's all about bringing the individual siRNAs at a low picomolar concentration. So one thing that you can, of course, do, and that you should do when you do an RNAi experiment, is use the lowest possible concentration. And it was interesting, when I started doing RNAi experiments in 2001, we actually used 100 nanomolar in HeLa cells. It was completely insane. Now here we ran a titration experiment. On the left side, you see a panel. We used these different genes, like I think it's like 12, and we used concentrations from 50 picomolar to 4 nanomolar. And here again, you see the knockdown. Here's no knockdown, 100% expression down to 0. And what you see that already at 50 picomolar, and that's using siPOOLs, pools of 30 siRNAs, right? What you see here is that already at 50 picomolar, you get a strong downregulation. And at 1 nanomolar, from almost all of them, you reached a saturating knockdown. There are a few where you may have to go a bit higher, but most are actually, you will not get better knockdown if you use more siRNA concentration. All you do is you increase off-target effects. So the message is use the lowest possible concentration that you can. On the right-hand side, you see similar analysis. Basically, we test a lot more siPOOLs, again, our reagents, and at different concentrations. So actually 700 at 1 nanomolar and 155 at 3 nanomolar, and a few at 250 picomolar. And what you see, the green curve, is the 250 picomolar curve, and you see that most of the tested siPOOLs actually already at 250 picomolar have reached like 70% silencing. And at 1 nanomolar, it's actually, I think, almost 90%, and most of them have reached 90% gene silencing. And then even going higher to 3 nanomolar doesn't really improve things so much. So again, for most siPOOLs in standard cell culture cells like HeLa, A549, and so on, 1 nanomolar will be sufficient. You have to use a bit more if you have, let's say, a very long assay, and of course you have to use more if you have a cell line that's very difficult to transfect. Of course, what matters is the amount of siRNA that's in the cells. But for easy to use siRNAs, for easy to transfect cell lines, 1 to 3 nanomolar will be perfectly sufficient. Which brings me to the fourth part. Now, we've been talking about off-target effects being a problem, and they are. However, they can also be useful. I've been showing you earlier that actually most of the effects that a single siRNA will produce is normally a seed effect, an off-target effect. So of course, that's a lot of information. It's just, as it's so degenerate, it's hard to make use of. But you can if you have large screening data sets. And this is actually something that we've been developing and that we are actually now offering as a service that we call PhenoVolt to people that have RNAi screens or that are running RNAi screens and that would like to have an additional seed-based analysis. And I would just like to show you one example. If you happen to have an RNAi screen and you would like to make sense of it, come talk to us, and it will be our pleasure to reanalyze your data set, and you may find interesting things. So I think, yeah. So this is an interesting case from a publication from 2010 that was done by the group of Norina Serial and the Max Planck Institute in Dresden. And they are interested in endocytosis. They ran an enormous screen with 100,000 siRNAs, and their assay was HeLa cells grown in multi-titer plates, and to which you add single fluorescence EGF and fluorescence transferrin. Each siRNA was transfected individually, and cells fixed and imaged by microscopy, by high resolution microscopy a few days later. then ran automated image analysis, and they extracted 50 features. And so for each siRNA, you actually ended up having a phenotypic profile that you can see here on the right-hand side. So 50 features on the different stainings, EGF uptake, transferrin uptake, and this nuclei, and yeah. So there's basically a specific profile for each siRNA. Now, what we've been doing is we've been comparing these phenotypic profiles of these 100,000 different siRNAs, and it's basically generating a correlation coefficient for each pair. The black line shows you a random pairing. So we're just randomly pairing these siRNAs and comparing their profile. And what you see here is a perfectly symmetrical Gauss curve where most of the siRNAs, basically most of the pairs have no correlation, which is what you would expect. Now, if you compare two siRNA, the profiles of two siRNAs that target the same gene, you get the green curve. And you see at the first glance that the green curve is almost random. This tiny bit that's shifted to the right side, that's the on-target effect. So just again, to show you how small the on-target effect is if you use single siRNAs, which is why it's so difficult, so dangerous to use single siRNAs. So then the red line shows you what happens if you compare siRNA profiles of two siRNAs that have the same seed sequence, and all of a sudden you see that most pairs that share the same seed sequence actually have a very good correlation, with a correlation coefficient of 0.6. So this is spectacular, and when we showed them, they were quite shocked, in fact. Now you can actually, now here comes the part that you can actually do something useful of it, and I will spare you the bioinformatics details of this. We have an excellent bioinformatician, Andrew Walsh, who came up with this algorithm that is actually useful when you have phenotypic profiles. There are other algorithms that you can use when you only have single readouts for each siRNA. So basically what it does is it looks at the potential seed targets and then basically generates a p-value for genes that are likely to be off target. And without showing you, like explaining any further detail of this algorithm, what you see is that here you get two enormous outliers. Two genes. One is called IDGAF, never heard of it. But the other one is a known one. It's called transferrin, TFRC. That's the transferrin receptor. And that, of course, is cool because remember, one of the readouts was fluorescent transferrin. So what that basically means is that most phenotypes were driven by an off-target effect on the transferrin receptor. So basically these RNAs, just as through the off-target effect, affect the transferrin receptors, and they just didn't notice. Now, interestingly, the other gene, ITGAF, this is a gene that wasn't even screened by any of the siRNAs. But as you go through the seed and do a seed-based analysis, your analysis is by definition genome-wide. So the cool thing is that with the seed analysis, even if your library did not cover the entire genome, if it's large enough, if it has decent and sufficient size, it will actually you can actually address each gene in the genome, and again can discover targets that you haven't even screened for. So that's actually a very powerful approach, and we're actually doing this mostly actually for industry clients who are pretty interested in this, because they can actually recover past RNAi screens. And that already brings me to the end of my seminar, and I would just like to give you a quick summary and again the contact details. So if I want to summarize this in 4 points. So transient gene silencing has multiple advantages over knockout in many cases, not always, but in many cases it will be the better approach. And the best way to get transient gene knockout is actually RNAi. So seed-based off-target effects are dominant in single siRNAs. So don't be misled if the single siRNA gives the phenotype that you would like to see. in high likelihood, what you see will be an off-target effect. So if you use single assignase, you have to use multiple assignase or use siPOOLs. So complex assignase pools minimize off-target effects by the seed dilution, as I was explaining you. And then, interestingly, the seed information can actually be used in large-scale RNAi datasets to find alternative targets that could not be found by on-target discovery. So the seed information can in fact be used. So if you're interested in RNAi, and if you need RNAi reagents, if you need advice on RNAi, if you need bioinformatics support, please contact us. We're happy to help. You can visit our website. You can send me a mail, and obviously also at our info address, or give me a call. And yeah, thank you for